Hi everyone, I'm Feargal. I'm coming to you from Flinders University in South Australia, and I'm going to talk to you today about a project that me and my team were sort of developing as a bit of a side project for the past year or so, and it's turned out, I think, to be very useful for us, and I'm hoping to present it here and everyone will use it and it'll maybe save all of you a bunch of time as well. I'm mostly going to discuss it in the context of human gut microbiome data today, but it's broadly applicable kind of across any type of metagenomic data, really. But a lot of the databases and public data sets are quite data orientated, as most of you probably are aware. So I'm going to begin by acknowledging that Flinders University is on the lands of the Ghana people, and that we acknowledge them as the traditional owners and custodians of their lands and their elders past, present, and emerging. So I'm starting today with basically this big table. So this is the output of a differential abundance analysis. Common test that a lot of us would do when we're analyzing metagenomic data. We'll end up with a table of species or taxa, genre maybe, and we'll have test statistics, fold changes, p-value, and maybe adjusted for multiple testing as well. And then we'll basically try to figure out, well, what does all of this mean? A standard way to do this is we'll cut it off at some point, maybe using our p-value or fold change threshold. We'll visualize the data, and then we'll dig into it in the literature. And okay, what do these changes mean? You'll talk to your colleagues, talk to a postdoc in the team, and have we seen this species before? Or what kind of metabolites do these produce? How can we interpret this data? How can we figure out our next step? Do we go validate this in the lab? Do we do another experiment? What's happening next? And it's quite time consuming, as anyone that's done it. I've done it quite a lot. And, over about a decade of doing it, you start learning names of things that come up over and over again. But it's challenging, it takes time. One of the really challenging parts of this is actually that the human microbiome in particular is very, very personalized. So in this heat plot here, I've visualized some data from the Human Microbiome Project. Each row is one species, each column is one sample, and then I've colored at the top what person they came from. This is just 8 people where there's longitudinal samples, just to capture how personalized that is. You see samples from each individual clustered together very, very nicely. But part of the problem with this, if we were, for example, looking at bacteria that were altered based on a treatment, so we want to know if certain bacteria will decrease in response to a certain treatment. Well, if we look here at this sample on the left, this dark green individual, maybe these bacteria will decrease in response to that treatment, but they're not in other people, so we won't see those decrease, which kind of means, the thing we're looking at an individual species level, that how true will that be when we carry it across different people, as we'll start seeing a personalized and different response. And this is just a general challenge when analyzing microbiome data, which is very, very sparse. In this table here, basically everywhere there's green, it's bacteria detected. Everywhere that's white, it's not detected. So how can we make sense of this, or how can we kind of deal with some of the sparsity or the personalized nature of microbiome data? Well, one thing that we've been doing for a very long time in this field is we'd simply aggregate counts to a higher taxonomic rank. So we wouldn't look at species, we'd add them all up and look at the genus, we'd look at the family, we'd look at a co-abundant group, something like that. This is useful, and you can definitely see some things with this, but you do lose resolution when you do this. You also risk missing shifts that can happen within a taxa. So maybe one species is susceptible to an antibiotic and another one is resistant. So if you just add that up to the genus level, that will cancel out. You could drop the taxonomic information entirely, move and look at something like pathways or gene family abundance in human 3. Very useful, very powerful. I use it all the time, but it does require a step up in terms of the amount of computational compute that you need. And it does generate a much higher dimensional data, which particularly for students or someone maybe that's stepping into microbiome analysis from another discipline, it is a steeper learning curve for sure. But of course, neither of these approaches will tell us if we're actually seeing something previously reported in the data. So I analyze a lot of multi-omics data across a lot of different contexts. And the first thing I always want to know is, have I reproduced any key findings from before? Because if I do, then that adds credibility to my novel findings. And other omics data kind of already have a solution around this. So looking at transcriptomics, RNA-seq, After we do a differential gene expression analysis, you would then contextualize your results using a gene set enrichment analysis. So we basically have these really big databases of sets of genes that represent some known biological function, some previously published association, and you can then see are these sets changed in a particular direction. This is really quick, just takes a few seconds, and then you're able to go into your deeper analyses, in that case, alternative splicing or network modeling, but with greater context, and you can kind of form a hypothesis to dig into that. If you see gene sets related to inflammation changing, then you might want to go and model some part of the inflammatory or innate immune related pathway in that data set, right? And so this is basically what we've done with a tool called TaxSEA. So TaxSEA is a gene set enrichment analysis style tool for microbial taxa. It allows you to test whether groups of microbes are enriched or depleted in your data. These groups can be from previously published databases. In TaxSEA, we bring in 5 published databases, 2 databases of metabolite producers, 2 databases of previously published associations, and 1 database of different bugs from different parts of human body sites. TaxSEA is very fast, it's very lightweight, you can run it on any laptop in just a couple of seconds, and it will help you kind of rapidly contextualize what you found in your data. I'm not going to dig into all of the features and benchmarking that we did. I'll link to the paper at the end, and you can read into that if you like. But basically, if we jump back to that table I showed you at the start, it would allow you to go from something like that to this, where this is the actual output of running this table, where we can detect a significant enrichment of oral and IBD-related taxa in this data. Now, this is an IBD data set, so it's pretty nice to be able to see that the oral-related taxa has a known signature in IBD. We can also see a significant enrichment of IBD and specifically Crohn's-related taxa here. Now, if I was trying to develop a diagnostic classifier based on this, I immediately have a red flag here because another disease signature has come up for liver cirrhosis. So that would be something to help me interpret how specific the alterations are seen in my context. The other thing that's important here, of course, is that if I'm analyzing this IBD data set, I can immediately see that I've been able to reproduce previously published findings. And so now I can go and explore my novel findings, such as metabolite producers. So if we were to plug this into the metabolite database, this is what we'd see. Enrichments of producers of certain neurotransmitters, decreases in producers of short-chain fatty acids. Again, these are known things in IBD, and I picked IBD in this context just because it is particularly well studied. But really, this sort of contextual or information previously would have relied on you digging into the literature for hours or days, whereas TaxSEA will provide this to you in just a few seconds. And you can visualize this in a few different ways. I love a volcano plot. So here we have a plot where each point is one individual bacterial species in this data set, fold change on the x-axis, p-value on the y-axis, and in the green, that's that shift of short-chain fatty acid producers decreasing in the disease data set, and then that increase in serotonin producers on the right. And you could imagine that certain members of these wouldn't be present in every person, but TaxSEA is able to detect kind of more modest or subtle changes that we maybe only see in subgroups, and then add them all up together to get that consistent signature. So TaxSEA was published in Briefings in Bioinformatics earlier this year. Preprint was out last year. It's available in Bioconductor, which you can use in R. If you don't like R, we also have a web portal that you can go and access this in, which is also able to produce some of those data visualizations as well. And with that, I will wrap up. Just acknowledge, so Kong and Tim are 2 of the students in my team that did a lot of this work. A lot of this was done by Callum Walsh at University of Melbourne. Me and Callum worked on this for a long time, and really core to our collaboration here was our 2 dogs, Oswald and Dexter, 2 very, very big dogs that enable me and Callum to take them out for walks and discuss how best to incorporate public databases into our microbiome data. So thank you very much, and look forward to discussion and questions.